Thank you everyone for the invite. Thank you for waking up on your last day just to listen to me. This is great. Unfortunately, this is going to be the voice you're going to hear for the next few hours, so I hope you don't mind my subtle Italian accent. I'm not even quite sure where I'm going to start, but let's start with what we do as a lab. So I'm based in Naples. I've been there for 5 years now. Before that, I was in the US for a number of years, and then I wanted to come back to Europe, but I overstepped a little bit. I ended up in Japan for 2 years. In Naples, we work on extremophiles, as Anais was saying, but with a little bit of twist on classic work that focuses on the physiology and adaptation of extremophile, and we look more for planetary scale effects of extremophiles. Why extremophiles? Not just because they're cool, but because they control a lot of the function on the planet. And it's one way for us to try to understand why we got where we got today, why this planet is different from other planets, why we have life here and we haven't seen it anywhere else yet, and what are the interactions. So as I was discussing with the rest of the instructor and the organizer what to talk about, I thought it was a good idea to give you not just a take on what is currently known about life emergence and evolution, but also why we do it and what are important features of life that we might use in our work. So before I get started, and you can read more about the projects we have ongoing and things we do on some of our website, I just want to know how many of you have a biological background? Raise of hand. Okay, about 1/3. How many of you have a geological background? Ooh, minority, big minority. Astro? Okay. Any engineers? Okay, we have a few also. Very nice. Did I miss anyone? Chemist, sorry. Wow, my apologies. Chemist. Any philosopher? So one of the cool things about astrobiology is that you come from all the different pathways of science. We don't have a discipline of astrobiology, but we have interest in astrobiology. And philosophy is one of the ones from where you can come into astrobiology. And so just as a reminder, astrobiology, and I'm sure everybody knows this, astrobiology studies the origin, early evolution, distribution, and future of life in the universe, which means not just outside this planet, but also on this planet. So that's why studies on the origin of life, studies on the evolution of life, studies of interaction of life on this planet and beyond. And is investigating deterministic conditions and contingent events, meaning what's happening by chance, and it's going to be difficult to be replicated anywhere else, what's actually driven by some of the basic physical-chemical law we know. And that's an interesting way of approaching even just if you don't want to look for life in other planets, but you're just thinking about life on this planet, this is a very nice framework to think about. Why we have life here? What would happen if we go back, we rewind the tape, and we play it again? Will we end up with the same type of life? Will we end up with humans after 4 billion years? Or will we have something completely different? There's one assumption here. The assumption is that life on Earth is not a cosmic unicum. It's not unique. That's the only underlying assumption in this discourse here. We assume that life is, I would say, fairly common most of the time when we have discussion. Now we're going to discuss about that later this morning when we talk about coevolution. But this is for sure one of the main premises when you start to approach astrobiology. Today, as I speak about extremophile and other things, I tend to jump. I'm going to try to avoid too hard jargon, but please raise your hand and ask if you don't understand any of the term I'm using or any of the thing I'm saying. Also, because my group works a lot at the interface between biology and geology, I tend to blur the boundary there. And since geologists are a minority here and biologists are not the majority here, I guess I'm going to maybe mix things up too much. So please stop me there. So if you think about theories, let's talk just for a second where we are today about this. There are a number of different theories about how life started on this planet and why, and it's actually pretty difficult to try to make sense of all the different theories. I'm going to just try to summarize where we are today, and many of you might have heard a portion of this before. So we have an extraterrestrial origin of life, we have a terrestrial origin of life. The actual difference between the two is that an extraterrestrial origin of life, in most cases, is not discussing how the origin of life happened. It just moves the problem somewhere else. How did life start on this planet? Well, it came to this planet, period. Done. Easy. So many people don't like to discuss this because even if you believe in an extraterrestrial, you're still faced with how life started somewhere else. So you can assume both. And then you have people that think about an organic or an inorganic origin. There's a lot of theories around clay organism and the role of clay may have played in the origin of life, not just as a place where organics started to concentrate and interact, but also actually as a template for basic processes to happen. And then people that think that organic origin could have used other organic as energy source, so things that may have been created abiotically, in the galaxy, in the cosmos, in the interplanetary disk, in comets, on planets, in several ways, or non-organic energy sources. And then as you go down, people believe about maybe exogenous delivery of organics, which we know was present, might have been the main source of things, endogenous production of organics, things like Fischer-Tropsch reaction or abiotic formation of organics, and this means that there was an heterotrophic life to start on, or the people that think about non-organic, they go both ways. Photosynthetic, chemosynthetic, some people say we start from inorganic, and then, very quickly one of the two takes over. And I found very interesting a strong push lately about photochemistry and the role the photochemistry might have played in starting this. There's another big division you don't see here, but you might have heard about, which is the division between people that believe metabolism first or information first. This is also a big one out there, right? People that think that protocatalytic metabolic networks started on their own, and then eventually we got control on this with information, and then eventually we got to enzymes. I know Joseph Moran was here. It's one of the people that is working on the possibility of creating complex self-controlling autocatalytic network of reaction, actually using metal as possible catalyst. On the other way, we have people that think that organics started self-organizing into information and enzymatic carrying molecules. And people really love RNA for this, because we know there's a catalytic aspect and an information aspect. Now, both have their supporting evidence, and both have dark spots. The reality is that whatever you choose, there's a long way from completely inorganic or a random soup of organics to any of these two, and then there's a long way from any of these two to any organism that we don't really understand. The third things that people discuss are scenarios, right? Where did life start and how? So you may have heard about the warm little pond, or the Miller-Urey experiments. And you know how this started as an idea of origin of life? How many of you heard about the term warm little pond before? So everybody knows comes from a letter from Charles Darwin that was saying to a colleague, oh, if we think about a warm little pond on the surface of planets concentrating all sorts of chemicals, and people took this like the Bible, went to town with it. Literally. So for many years, this has been the main theory for origin of life. And then, of course, you heard about hydrothermal vents, origin of life, especially alkaline vents, origin of life. The idea that this porous material with alkaline fluids created natural hydrogen proton gradient across these porous media, and then the porous space inside could act as protocell, concentrating the organics and allowing for life to start. Then there's a third popular one, I'll bet much less than these two, which is called the nuclear geyser. I don't know how many of you heard about this one. This is a Japanese theory that has been worked a lot in the past 20 years. The idea is that you get radioactive decay, ionizing radiation. This might create hydrogen from radiolysis. Hydrogen is a powerful electron donor. It can help with the creation of organics, and that's how things got created and eventually connected to the surface. And there are other theories that are less structured, perhaps. The role of photochemistry in many of these, both up here and down here. The possibility that it started into sedimentary rocks like clay and so on. Now, people assume that because I work on hydrothermal vents, I must subscribe to the hydrothermal vent origin of life. And if you ask people, everybody usually asks their favorite. Now, my take on scenario is that, and it might be a bit unpopular compared to many astrobiologists, scenarios are very useful as long as they help you discover something about principle that control the processes we are studying. Otherwise, we have no evidence to support one or the other. Not really, because there are counter-arguments everywhere. And I think that a lot of time our field steps into more philosophical, faith-like things when they say, oh, it must have started here or it must have started there. Because we have no evidence to support it. It's more like a belief that we have. So whatever your thinking is in terms of scenario, I think all of these are very important to study fundamental processes. But short of a time machine, it's going to be difficult for us to know where life started. And life may have started in multiple places. We'll talk more about that later. So it becomes very, very difficult. But that's a very popular question I get all the time. Where do you think life started? Well, I don't know. I have no idea. And I do work on the origin and evolution of life, mainly on the emergence and evolution rather than the strict origin. But my answer will be, I have no idea. I take a very simple view, simplistic view about what life is. Somebody said that you can have as many definitions of life as are the researchers working in origin of life or in biology. And I take a very simplistic view on it, mainly because you need to answer question on why life is there and on its thermodynamic and entropy relationship with the law of thermodynamics, with the planet, with chemical gradients and everything else. And I really like this definition here. Life is nothing but an electron looking for a place to rest. Now, this is very simplified, but in a way, it's telling you why a planet might need life to emerge. So I thought, since we have a very diverse background, as we've seen, and biologists are, despite calling it astrobiology, because everybody's got to focus on life, biology is not the main discipline, and you're very well representing the field. I thought I might share with you how I see life and some of its fundamental property, and how this, in turn, might change the way you approach some of your science or you search for life. The emergence of life here or life somewhere else in the universe. So the first one, you may have heard it. Life is electric. I don't know how many of you heard that, but I mean this literally. Like whatever energy form you choose for life, you're thinking about phototrophy, you think about chemolithotrophy, you think about heterotrophy. So you have light as an energy source, you have chemicals as an energy source. At the end of the day, everything is about redox chemistry. Whatever energy form is a redox reaction. Life is based on redox. And so in photosynthesis, you're using light to strip electron from water. In chemosynthesis, you're good to strip electron directly from reduced compound like this one. And you can see they're actually water and hydrogen sulfide are not that far off in a lot of ways. So life is about redox. And it's not just about that. Life is literally electric because it does what a battery do. You know, biggest invention of Alessandro Volta, the battery, was to put two metals that have a redox reaction close to each other but not in contact, separate them with a place where ion can flow but electron cannot, and then connect them with a wire on the outside that is just carrying the electron around. So basically what Alessandro Volta did was take a process that is a scalar, that has no directionality, and for the way he arranged the system, he got a vectorial process that's direction. And you know, engineer, physicist, when you have a vector, you can do work. Huge invention. Biology does exactly the same. Many people will tell you that life's greatest invention was the membrane, because it can separate the inside from the outside, concentrate things, the self from non-self. To me, the biggest invention of life is the membrane, but just because it allows you to transform a scalar process into a vectorial one. So you have a redox couple, and you are separating where the protons are going outside from where the electrons are going across the membrane. And because you're separating in space and time the two redox reactions, the two alpha reactions, the life needs to get energy and do work, now you can do work. Literally, life is acting as a battery, pushing electron one way and protons the other way. When things happen in the same place, you cannot control the reaction. You cannot do work where you're able to split the two sides and decouple them in time and space, and sometimes even between different organisms. More technical, but we have organisms that share half redox reaction across space, like methane oxidizers with sulfate reducers. Now you can do work. So to me, life is electric in a very literal sense. It's not a metaphor like when people talk about neuron and things like that. The second one you should know is that because you want to control redox chemistry, to do it, you need to find a way to shuffle electron in a good way. And the way that life does it is using metals. Now, if you think about popular gases, whether you work on climate, you work on atmospheric atmosphere composition and detection, whether you work on normal redox metabolism, whether you think about origin of life, methane, nitrous oxide, CO2 are very important gases. Now, these are both produced and consumed by life all the time using fairly complex molecules. Now, I don't want you to think about these molecules and their names and their structure and their multimeric nature, but think that the center where the redox chemistry of these gases is happening is actually a metal. So they're using nickel to produce methane. They're using copper, nitrous oxide, iron sulfur cluster for CO2, and many, many, many other metals. So as a non-biologist, when you think about redox protein, which is a subclass of proteins that are controlling redox chemistry, that I just told you is at the basic of whatever energy life is using, you should think about a very fancy assembly Lego that can keep actually a number of metals close to each other to allow electron percolation to happen. So basically you are keeping small conductive pieces that are tuned exactly to the right midpoint potential for electron to hop from a donor molecule or a donor chemical compound or an element going through your chain and going somewhere else. And life is all about controlling this. And proteins are just a fancy way to keep these metal centers close to each other. And when I say close, I mean usually 14 to 20 angstrom, because that's the perfect distance for electron tunneling, electron to jump around in a specific way. So as a non-biologist, redox protein, a special class of protein for you, should be just a fancy way to get electron in the right place. It's like literally as an electrical circuit. The other thing that is interesting, probably especially for the chemist in the room, is that the diversity, not just of metals, but metal organic structure that life uses, it's amazing and it's incredible. And if you think about our current modern chemistry, where organometallic catalysis is very, very important for a lot of the things we do, it's striking to see the diversity of structure that life has created, evolution has created over time to control different redox chemistries. And this is a very incomplete picture that we have, because right now we know organometallic cofactor structure only for microbes we can culture in the lab. And we can culture in the lab a few % to 10% or 20 depending on the environment we are. So this is striking because there's a huge potential for biotechnology, huge potential for discovery, huge potential for controlling any type of selectivity of reaction out there. And we have no idea. The other thing you probably don't know, one more thing, sorry. Every one of these structures appears to have some type of roots in geology. So there's a direct connection. If you look, for example, for the nickel substituted graphite here, and you look for the cluster inside carbon monoxide dehydrogenase, they're virtually identical with small deformation. And what we think happened is that there must have been a point in time in which minerals were catalyzing a biotic reaction. And we know that's true in many ways, and we can still reproduce it. So some of these must have been appropriated by biology at some point. So maybe on mineral surfaces, or in solution, or in nanoparticles. And then eventually we got to biological control of the synthesis of those compounds. And that's why life can interact with minerals in many, many ways. Another thing you might not know is just how many elements can life interact with. Did you ever ask that question yourself? If I take the periodic table, how many elements can life interact with? Well, we know carbon, hydrogen, phosphorus, nitrogen, sulfur, and so on. But have you ever tried to do the child game of coloring the periodic table? No? How many? Nobody? There are some serious astrobiologists trying to do this. And it's not a trivial exercise, because once you start to do it, you realize that it's actually faster to count what remains blank than what is used by life. And I mean this. This is an incredible SWOT of the periodic table. So you know that life can breathe oxygen. Breathing means using this molecule to dump the electron at the end. Is your sink at the end of whatever metabolically redox you're doing. We breathe oxygen. You might have heard that life is breathing nitrate. You might have heard that life can breathe sulfate. But life can breathe also arsenate, selenate, tellurate, uranate. The list is very, very, very long. And then once you start to look at this, you might also realize that this is not a complete picture, because we are still discovering elements that life is using. For example, in the last 8 to 10 years, we discovered enzymes that are very important for the oxidation of methane present in the atmosphere that are actually lanthanum, cerium, and niobium dependent. So they use the rare earth elements that we always believed were completely inert with respect to life. They're actually being used as key cofactor in enzymes. And if you remove the lanthanum and you put the calcium, the enzyme just doesn't work that well. And discoveries like this are happening all the time. So even the periodic table, which is a very finite number of elements, we still have no idea how much interaction is out there. And all the implication you can think about search of life, type of life, metabolism, biotechnology, ecology, you name it. And because of this, they interact with minerals a lot. Now we have 3 examples here: amorphous calcite, pyrite, iron sulfur framboids here, and iron hydroxide twisted stalks. Now many of these, like these two, are prime indicators of life interacting with minerals. But the list is actually very long. We have hundreds of minerals we know that life can either dissolve or precipitate. But at the same time, we know these only from microorganisms that we can culture in the lab, which means that we still have an incomplete picture of the interaction between biology and geology, at the interface in which life can interact with the solid planet. Very incomplete list of this. The other thing that struck me as a student was the diversity of redox reaction. So I was always looking at the redox reaction that we write when we isolate new extremophiles and new microbes, and it always felt like a bit incomplete. And I have a colleague, a very smart colleague, Everett Schock, Arizona State University, that sat down one day and wrote all the possible redox reactions which have a negative delta G, so they're spontaneous under certain conditions on this planet, that biology could use for getting energy. And he limited himself just to carbon, hydrogen, nitrogen, oxygen, sulfur, and a couple of minerals of iron. No tellurate, no uranate, no arsenate, no any of the other fancy stuff, just the basic, very basics. And he was able to write 1,500 reactions, pretty much. There's 1,500 redox reactions that could sustain life. Pretty amazing list, because if you just add one more elements, the combinatorics explodes, you go up to 3,000, and then to 5,600, and then to 12,000 reactions, and things get out of hand pretty quickly. Now, the other very basic question, and I like science because sometimes you can ask the most basic textbook question, and you realize that we don't have an answer, is how many of these reactions are actually known to be present in life? Because this is a theoretical list. Do we know exactly which reaction is used? Do we know which one is used by who? In other words, do we know the distribution of the tree of life? We take a tree of life. Do we know exactly where things are? Well, the reality is no, we don't. We have no idea how many of these reactions are existing in the environment. We have no idea how they're distributed in the environment. Because in many cases, even when we grow microbes in the lab, we don't do the painstaking, long, old-school job of measuring all the possible input and all the possible output of that organism. And these are huge implications. For example, if you're looking for atmospheric composition, we have no idea what microbes are getting in and out. How can we model what's happening? And so a very basic question like how many of these reactions are being used, we have no answer for. Two more things that I think are crucial for biologists and for everybody that is not a biologist to understand is that at the end of the day, and this is Everett Schock again, biology does what geochemistry allows. So which means that in energy term, and for the majority of the reaction actually, biochemistry, what life does, is a subset of all possible organic chemistry, given the specific boundaries of the planet. In other words, biology works with whatever the planet provides. Now, there are caveats we're going to discuss later on this morning when we talk about interaction and coevolution, but in general, you should think about biology is able to do whatever the planets allow. And there's one more thing to know, is that biology likes it slow. Or the way Everett says it is that things that burns into flame are not good to eat. Now, let's clarify this concept here, because I think it's one of the fundamental concepts in biology. You know sugar, right? Carbohydrate. It's carbon, hydrogen, and some molecule of oxygen. And you know the sugar can burn. Have you ever tried? Take powdered sugar and try to set it on fire. It burns very, very well. Carbohydrate can burn so much that I don't know if you ever heard about flour explosion. In place where they do milling of grain to make flour, if it gets into the air in a very specific concentration, ratio air to flour, the entire plant can explode completely. It's actually very dangerous. Why? Because carbohydrate, they react with oxygen, freeing a lot of energy. That's what we do in our body, right? We burn glucose with oxygen to make a lot of energy. So this is a spontaneous reaction. And yet, if you leave sugar on the table, it's not going to catch on fire on its own, right? Because thermodynamics is not everything. There's activation energy and there's kinetics. So life likes it when you have a negative delta G, the reaction is spontaneous, but things are not bursting into flame on their own. Otherwise, it cannot use it. It's an uncontrollable energetic reaction. There's nothing it can do with it. So life is very good at speeding up things that are already slow. Life is very good at using free food that is not burning already. These two concepts together, they might very well explain all the geo-bio interaction out there, all the biogeochemistry, all the geochemical anomalies we see, and all we expect to see in other planets. But these two concepts are somehow often misunderstood. And so we're going to spend some time discussing them in a little more details. By the way, a lot of these ideas are in this paper here by Everett Schock and Eric Boyd, Principle of Geobiochemistry. They inverted the term. You might be used to hear biogeochemistry. But because life does what geology allows, geology comes first, life comes later, so it should be geobiochemistry. That's one of the arguments in this paper. It's a very general paper, good for everybody, not just biology, so I really suggest you read it. So we said microbes like it slow and mostly free, and they want to make it fast. So they do this in two different ways. One is by lowering activation energy to doing reaction. Think about hydrogen and oxygen. We use them to send rockets to space. And there's biology on this. And we know that it's very dangerous to have hydrogen and oxygen together. You just need a spark and things go boom very quickly. And yet there are microbes that can actually use hydrogen and oxygen as a fantastic redox couple. They use oxygen at lower partial pressure, so it's not as dangerous if it starts, and they're able to control this reaction very nice because there is an activation energy there. There's a barrier that they're very good at catalyzing and overcoming. And the other one is that they like to do things faster than abiotically. And usually they're also good at moving equilibrium in some way. So the reaction equilibrium, they're good at moving it where they like it. I want to make you an example to follow this a little better. Iron 2 plus oxygen, we know iron rusts. This is a fact of life. If you have iron around and there's oxygen, it's going to make rust. So this is exactly this reaction. Iron 2 plus water makes iron hydroxide. If there's water around, it's going to rust, precipitate, makes wonderful orange colors. Now, we know this is a spontaneous abiotic reaction. But you should also know that there's a good amount of life that can use this reaction as a redox couple to make energy. They're called iron oxidizers. I know we lack imagination in microbiology, but that's how we call them. Now, the interesting fact about this reaction and many reactions out there is that even when you are within the domain of life, the abiotic reaction and the biological reaction coexist in the space, okay? And the relative contribution of those two to the total reaction that you are seeing changes depending on the environmental parameters. So you might be in a place where there's a lot of abiotic and very little biology contributing, or you might be in a place where the conditions allow biology to do everything, because the abiotic reaction is so slow and the kinetics are so slow that they don't contribute much. This is one of these fantastic reactions. So if you go to springs in the Italian Alps, that's what this group did, as always, average shock group, they do amazing work, and you go across spring with different pH, and you start to do experiments to measure exactly how much of this iron oxidation I see is abiotic, how much is biological, you see that the reaction is pH dependent. And this is the log K, so as pH goes up, the reaction gets faster. And it gets faster both for biology in red and abiotic in red. But then as you turn around 7.4, 7.5, I don't see red dots anymore. You don't measure. Red dots are down here, are somewhere inexistent. They disappear. What happens there is that the abiotic reaction becomes so fast, the biology gives up completely. And so if you look for the contribution of biology to the total reaction, if you are acidic, biology does 90 to 100% of the reaction. Biology is completely controlling iron oxidation and acidic pH. But as you move in pH, biology is struggling to keep up with the abiotic rate until they do about 50-50, around 7.4, and then biology gives up completely. It's the same redox reaction, it's the same delta G, it's the same amount of energy, it just cannot compete. And so you go from having a 90% biotic reaction to 0 biotic reaction, because biology likes it slow. And this brings us to another, this is a hint to me on the why we have biology on the planet. We know things physically need to be dissipated, right? We're moving toward maximum entropy. And as a planet is cooling down, it's trying to dissipate thermal gradient, chemical gradients, everything. But when the planet cools too much, things get trapped. There's a lot of chemical redox energy trapped in the planet. And biology is very good at speeding up when geology is inefficient. Like in the oxidation of copper sulfide, chalcopyrite, which is a prime mineral used to extract copper globally. About 30% of the copper we use every day in our circuits is extracted by microbes. We didn't know that for many years, for thousands of years probably. We know now it's extracted by microbes. Why? Because they can do the oxidation of the sulfide mineral 1,000,000 times faster than the abiotic reaction can. And so there's a lot more copper leaching out of the crushed rock if biology is interacting than if you just pump oxygen in it. So biology likes it slow and is very good at making it much faster. But the relative contribution of the two abiotic to biotic reaction is going to change depending on the condition. So if you think biology is if you think biology can contribute to whatever signal or reaction you are looking for, and thermodynamics is telling you there's a lot of energy for biology to use, don't just assume biology is doing it, because it's going to depend on the kinetics and activation energy, which cannot be predicted alone by thermodynamics. So we need a lot more work on this that we are not doing. For life to make a living, the abiotic rates needs to be slower than the biotic rates. I think this is a pretty universal rule about biology. By using all that, interacting with a lot of elements, interacting with a lot of redox couple, being able to speed up things, how much and how fast did life colonize this planet? So some time ago, with some colleagues, we started asking ourselves, what are the boundary conditions of life on our planet? So because water seems to be one of the major controlling parameters of life, because if it's completely frozen, you can't do things, and if you're breaking hydrogen bond, things are not stable, so you need to be in a space where water is liquid and not boiling or not frozen. We started asking, where do we find liquid water on this planet? So we started searching, and it wasn't easy to see the pressure range, pH range, temperature range, and salinity range in which we still have water activity. For example, this might look strange, this high number here, but we know that in deep sea hydrothermal vents, because of the pressure, water is boiling at higher temperature, so you might have liquid water until quite high. And the same goes for all the parameters you see here. And then the next exercise was to say, okay, let's take everything we know about life and let's map it on top. In other words, the state space of variable, physical, chemical variable controlling water, how does it map to known life on this planet? And this was the answer. Now you see, life apparently has occupied all possible ecological niche, broadly speaking, physiochemical niche, let's call them, on this planet, all of them. And even beyond, look at this. Oh, very low pressure here. Why? Well, because we're exposing stuff in space, and we know they can survive very low pressure of space, which is not found on Earth, technically. It's found near Earth, so we found things that were a lower pressure than we were expecting. But for everything else, you can see that things are actually very widespread on this planet. Now, how many of you know the highest temperature? Biologists, you know it, don't mention. What's the highest temperature at which life can be found on this planet? Polite guesses. Come on, you had a cup of coffee this morning, I guess? 120? 122, yes. But that's the current high temperature. The upper temperature limit of life, like every other limit of life, has been a moving target for the past 100 or so years. We started off thinking that no life could survive above 60. That's why we're doing pasteurization of things, because they were working with pathogens, and they knew if you expose a 65 for long enough, pathogen die. And so that was assumed to be the top limit of life. And then somebody said, no, we cultivate the stuff at 75, so the limit must be 80. And then somebody, a guy called Karl Stetter, a German microbiologist, which enjoyed, like many people, coming to Italy for vacation. At the end of every one of his vacations in the Aeolian Island in Sicily and in Campania near the Vesuvio, was filling up bottles of boiling muds near the beach and going back home and working on it. And he isolated things that grow at 85 and 90, 95, 98, 99, and everybody was sure that the limit is 100, right? Why 100? Because water boils. Well, no, if you are under pressure. And a few years later, he isolated the microbes from 10 meters depth in the sea, shallow water hydrothermal vents, where the upper temperature was 103. That made headlines everywhere. First life above the boiling point of water. Well, technically not, because you are at pressure, but let's say, call it that way. And then we kept moving it. 10, 13, 15, 19, 21, 22, and now it's pretty much a pissing contest to whoever can get to the highest temperature. But the same is for the lowest one. You have any idea what's the lowest one we can get off today? What's the lowest temperature which life is still active and we can cultivate it? Minus 18. Some of these, we tend to think in isolation, minus 18, 122, but actually the space at which life can survive is given by the combination of the different factors. We just said you can be at 122 if you have high pressure, because water is not boiling. You can be at low temperature if you have salt, because water is not freezing. So we don't speak a lot about single extremophile, but we call them polyextremophiles, because they need an interaction of extreme to survive. And this one here, we put it together 5 years ago, wow. It's the current record holder in different things, but I'm pretty sure they might have changed it by now, and they will change in the future. And for example, we can go from negative pH to 12.5. We can go all the way to 110, I didn't put pressure here. We can go all the way to 135 megapascal, but we exposed some microbe to gigapascal here in France, in Paris, and they still survived. So we don't know the number of elements life is interacting with. We don't know how many redox reaction life is able to support, and we don't know where they are the true limit of life yet, because it's a moving target. And many people in my field, working in subsurface environment and things like that, we believe that life might live up to 135, maybe 145 degrees in natural ecosystem. We are not sure, because 122 is the limit in the lab. So we think in the environment there might be something more. Some people put the limit at 135 because they know the phosphate bond in ATP breaks down. It's a good argument. But before the limit was at 95, because DNA denaturates, and we know there are ways not to do that. And then the limit was 100 because you break hydrogen bond, and there are ways not to do that. So I will not bet my hand that 135 is the real limit. We might be surprised in the future. Life interacts with all scales of the planet. Many of you might think of life as something that is actually very, very fast. It's not necessarily fast. We are fast. Pathogens are fast. Some animals out there are fast. We know trees that can live thousands of years. That's already pretty surprising. Well, we have a calculation that suggests that microbes in subsurface may have a doubling time. They reproduce their entire population every 1,000 to 10,000 to 100,000 years, if at all. Which means that they're working on completely different time scale than we are. Which means that a lot of the biological effect in blue, both on temporal and spatial scale, they actually overlap very, very well with geological scale. And so the potential for life to impact the planet is huge. We will talk later this morning, is not just oxygen and GOE and atmospheric composition. There's a lot more going on in this interaction that sometimes we're overlooking. And these are the doubling time calculation going down inside sediments. And you can see they go up quickly. And this is a very short read, like 2 pages from Karen Lloyd, Time as a Microbial Resource. If you never thought about time as being something, a variable for biology to interact with, well, I invite you to read this paper. It's a very interesting one. The thing you probably already know, so we can go a little faster, is that this is a microbial planet. And this has been a microbial planet for very long. Have you seen this one before, right? The clock of life on this planet. So the planet starts up here, 4.6, 4.55 billion years, then around 3.8 to 4 we get the first earliest life form. You keep going, keep going, keep going. You get eukaryotes, still unicellular, and then somewhere around here they start to be multicellular life and so on. And humans are just a tiny dot up here. Now, if you go through this, you see that these, out of 4 billion years of life on this planet, anywhere from 3 to 3.3 billion years, this was only unicellular. So this has been a microbial planet for a long time. And it still is a microbial planet. As microbes dominate the bio in biogeochemistry, is almost exclusively microbial. Carbon cycle, very strong component in biology. Nitrogen cycle, almost exclusively biological. You can include human in the biological with our fertilizers and fixing N2 into nitrate and things like that. Sulfur cycle, strong biological. Oxygen, strong biological. Iron, not that great. But if you do some calculation, more than 45% of oxygen is done by microbes, microorganisms. More than 45% of primary productivity everywhere, probably a lot more if you account for chemosynthesis. 90% of organic matter recycling, 100% of nitrogen fixation is biological. Now we've been altering that for the past close to 100 years. The methane emission are almost exclusively microbial, and so on, and so on, and so on. So this has been a microbial planet and is a microbial planet. Now, an interesting thing that came out of the last 30 years of research is that we discovered that life is not just on the surface of the planet, but is also on the subsurface. How many of you have heard about subsurface life? Okay, good amount. Amazing. Well, the idea doesn't come from a biologist. Like many good ideas in biology, they come from somewhere else. And in this case, it was a geochemist that proposed the idea. The same strange geochemist that believed that petroleum was abiotically formed in the crust all the time proposed that perhaps in the subsurface there are deep, hot areas where biology is present. This was only in 1992 when this idea was set forth. And in the last 30 years, we discovered that it was very right, and not just where it's hot. But there's a lot of biology down there. So much so that if you do a carbon balance of this planet, a lot of carbon is terrestrial. Why is terrestrial a lot of carbon? Why is it on land and not in the ocean, a lot of carbon? What's the biggest storage of carbon on the surface of the planet? Organic carbon. Cellulose, plants, wood, things that are recalcitrant, things that cannot be degraded easily. That's a great place to put carbon. The ocean is only 1% of the total biomass, or less. And look at this. This giant here, 13+% is actually in the subsurface. So the subsurface, technically speaking, contains the second largest reservoir of carbon everywhere. But compared to the green one, terrestrial, where it's all plant and fungi and protists and animals, look at the subsurface. It's all bacteria and archaea. Of course, it's all microbial. So we have a huge amount of microbes that are below our feet and below the ocean floor, everywhere. And we know that they go pretty deep. These are cell counts in different locations. And right now, we know that we have counted cells in sediments or cores down to 3.5 kilometers in the ocean and down to 5 kilometers on the terrestrial crust. And they keep going. And there are calculations that show that if you assume 122 as your top limit, life might go as deep as 10, 15, or 20 kilometers into the crust. This is a gigantic portion of our planet that is completely filled with bacteria and archaea, with a lot of effects that we're going to discuss later. The last one, which is going to be what we're going to discuss later on, is that life and planet have actually co-evolved a lot more than you think. Now, there's a huge misunderstanding out there about how this works, and it's changing in the last 5 to 10 years, I would say. But since I'm very interested about this topic, I spent some time during some of the conference we were at asking questions to people. So I started going to people, random, because I'm very shy, and asked to people, are you in the geosphere or biosphere? Where do you identify yourself? And bear with me, because you can ask this question also to chemists, depending on how they approach things. If they think they're closer to the geology and the geochemistry, or closer to the effect in biology, or at least at conference where I go, people tend to have one of these two camps. And I said, okay, you're a geologist, or you're a geoscientist, or earth scientist, let's call it the way you want. Tell me what are the 10 most important moments in the evolution of our planet. Now, I know we have only 3 or 4 geologists in the room, but as I say this, you are probably thinking, and probably everybody else is thinking, what are the most important moments on the planet? Well, after doing about 30, 35 interviews to geoscience people, I got this. So we have earth formation, core differentiation, the core is important on this planet, and then we have moon forming impact, late bombardment with some debate if it really happened or not, and then we have the Wilson cycle, which is actually the onset of our modern plate tectonic, the great oxidation event, snowball Earth, and then we get finally to modern Earth. So this will be, many people put other details, but this will be a consensus of what the timeline of our planet looks like. And then I went to the biologist people, and I asked the exact same question. Now, there are more biologists in this room. Anybody cares to try to tell me what are the most important evolutionary moments for our planet? I hear whispering, but I cannot understand. No? It was very interesting because I got this. And the entire top timeline is actually formation of Earth, 1 point. So for biologists, everything there happened in 4.5 billion years is 1 point. But we have first evidence of life, we have stromatolites, rise in atmospheric oxygen, first nuclei, multicellular life, the first land plant, first animals, and human. These are 2 completely disconnected ways of seeing the evolution of our planet, except for one spot. Invention of photosynthesis, great oxidation event, right? This is probably the most well-studied geo-biofeedback co-evolutionary moment on this planet. And even if it's the most well-studied, we still haven't understood much about it. And we can keep discussing this for years. And there are aspects of this that are actually often omitted when you read it in textbook, when we discuss it, and so on. So we're going to spend some time this afternoon about this. The key point here, and we're going to delve into it later, is that there's a lot more interaction across this entire timeline than whatever your background is telling you to. And this is just because we are not used to think from the other side of the camp. We are not used to think in a very interdisciplinary way. We are set in our ways, we learn our textbook, which have a one way to see things, and we lose the perspective on the other things. Now that you all agree, I'm sure, about the way I see life on this planet, and I shared with you a little bit of some of the interesting things. I think everybody should relate a little bit, even if you're not a biologist, because I think they have implication. We're going to discuss about the implication later. How did we get here as biology? So we hear a lot about talk about origin of life, and you all heard about Luca, right? The last universal common ancestor. Well, there's a lot that goes on between there and whatever happened before. Generally speaking, we are using two different approaches to try to understand more this. First one is a top-down approach. We start from modern life and we try to work backward. All the molecular phylogenies, the trying to break down the minimal set of genes, trying to break down the minimal portion of any enzymes that can do things and things like that. This will be a top-down approach. Now, what's the problem here is that, well, it's easy to conflate historical genesis with modern utility. What does it mean that? Well, ribosomes are everywhere right now, in all known form of life. And we could conclude that therefore they were very, very important for the origin of life. But that's not necessarily the case. The fact that they're here everywhere now solving a problem doesn't make them the way life solved that problem early on. So this is a caveat to keep in mind. Do not conflate historical genesis with modern utility. That's something I do all the time, and everybody does all the time in this field. The other thing is that we have bottom-up approach, starting from minimal set of chemicals or molecules or genes or whatever, starting to build life up from the bottom. This is what Joseph is doing, for example, and many others in the field. And we're doing tremendous advances in the way we are understanding how you can make the building block of life this way. It might provide clue on possible processes and pathway, but not necessarily linked to historical genesis. And I mean something very specific here, we're going to discuss in a second. We may find a lot of ways to do things, we're never going to know how things were made early on. So often with my people, we joke we should not call it Luca, we should call it Lux, the last universal common survivor. Why? Because most probably this is not a tree in the way we've seen it so far. This is more like a net. You all heard about that, hopefully. How many of you heard that there should be the net of life and not the tree of life? Okay, maybe we need to talk about this just a little bit. The point is that whenever we had the Luca, what we call Luca, the last common ancestor, this is just a long way away from the origin of life. And we might have had a lot of other branches that disappeared. And one of the things is that at one point or another we lost all these other branches, and now it looks like we all have a common origin, or we all have a common ancestor, which does not mean we had a common origin. In another way of saying it, at the same time, whatever bro has to go from origin of life through Luca, through modern Earth life, might look completely different depending on the condition. And in other places in the cosmos, even if we find how life originated here on this planet, you might have taken very different path because the environment is different, right? Because biology does what geochemistry allows. You do what the planet allows you to do. So the path to life might have looked completely different in other places. It's not that because we understand how things happen here, we're going to understand how things happen somewhere else in the universe. And currently, we have these three alternative hypotheses for the origin of life that we cannot really distinguish. First one is a single origin of life that split into branches. Some have gone lost, the early extinction, which we don't know anything about. And then we got Luca. But we might also had independent origin of life, and something got lost on the way and never survived or mixed. Or we might have had independent origin of life that merged at one point or interacted and mixed at one point, and we still might not be able to distinguish. So from where we're standing right now, single origin of life, multiple origin of life, interaction between different origin of life, they're all equally plausible. All of them. And we cannot know which one is the right one. The other thing you should always keep in mind when people speak about Luca. Luca is a complex organism. It's a modern-like organism in many ways. There's membrane, there's ATP, there's ribosome, there's DNA, there's RNA, there's a lot of stuff. From there to the origin of life, somewhere into prebiotic chemistry, major innovation, you cross what we call necessity contingency boundary, and you keep going until you fully alive, and we don't really know what that is yet, but let's call it fully alive, there is a very, very long way from one to the other. A very long way of some things we assume they should have happened because of modern necessity, or we must have had a phospholipid layer. We needed a membrane. We needed an ATP synthase. We need a ribosome. We need DNA. But in reality, that's conflating modern utility with historical genesis. And we have a huge gap of things that we're not sure what they are, or how they happen, or in one order. So I would say, Luca, let's take it for granted, Luca. Everything that happened from up to here is still up for grab in terms of research. Everything needs to be done in this space. We do know that after Luca, things got interesting. You ever heard about the endosymbiotic theory? How many of you? Okay, not many. Well, the endosymbiotic theory is a theory that started in early 900 with Russian, and then Lynn Margulis developed in the 60s. It's a theory about how eukaryotic cell, the cell we are made, and animals are made, the plants are made, came to be. Because eukaryotic cells are strange. You have a membrane that contains things inside that have other membranes. For example, the nuclei. Me, you, and all the other animals in this area, we contain another set of organelles inside a membrane called mitochondria, which is where we burn the sugar with oxygen. And plants out there, they have another cell of organelle with their own membrane that are called chloroplast, where photosynthesis is. And it's not clear where things came to be, but the theory developed, and we have a lot of evidence for this, tells us that mitochondria and chloroplast were actually once free-living bacteria. There were bacteria in the environment that got captured inside, brought inside by another cell, and they became what they are now. And we have a lot of evidence. You can extract DNA from mitochondria and chloroplast, and if you do phylogeny, they fall inside the bacterial tree in two very specific places. Not only we know that, let's see what I have. Here. We know that mitochondria and chloroplasts are not made by the nuclei, built. They only come from pre-existing one. So the mitochondria you have in you right now, they were given by your mother, by direct binary division, the same way bacteria divide. And chloroplasts out there, they come out in the same way. The nuclei cannot make them. They need to divide in the same way bacteria divide. Their DNA, they have DNA inside, they have their own genome, is circular, has no histons, and a lot of other evidence that becomes more and more biological and technical that tells us that these guys were once free-living organisms. So what we think happened is that there was some type of cell that people call proto-eukaryote that at one point or another had the nuclei. This is one of the biggest mysteries right now in biology. Where does the nuclei come from? Is it another being that was captured? Was it part of the original proto-eukaryotes? We don't know. And then at one point, we got a symbiosis going with the aerobic bacterium that was inglobated, and that became the mitochondria, and is a bacterium that is able to burn sugar with oxygen. And then in another symbiotic event, we had a chloroplast happening, and we have modern photosynthetic eukaryotes. Now, if you are interested in this, it gets more exciting, because then this guy inglobated one of these guys and gave rise to some protist. And then the guy that inglobated this guy inside had one of these inglobating one of these. So it's like an animal keeping a plant cell inside, getting another piece of an animal. You have up to triple endosymbiosis. And a lot of the strange animals that you can find in the waters of these ponds, euglenids, dinoflagellates, things like that, are actually endosymbiont of endosymbiont of endosymbionts. So we have evidence that this happened multiple times during the evolution of life, not just early on. Now the mystery here that people have been working on for the past maybe 60 years now is where these come from. We know this, where is this coming from? Now there are a lot of alternative evidence, alternative suggestion. In the 70s, Carl Woese started using 16S, which is a specific genes that is present in all life, exactly because there are mitochondria and chloroplasts, you find it everywhere, to just relate life to each other. In doing this, he found a new form of life, which today we call archaea, in Yellowstone. They were not similar to anything he's seen before. It was branching very far away from everything else. So he renamed things. He started calling the bacteria eubacteria, the real bacteria. And the archaea bacteria, the old bacteria, because they were in hot pool, boiling, acidic, they reminded him of early Earth. Then we changed the name again going forward. But the point is that at one point we found two domains of life, bacteria and archaea, that were different from each other. But both of them had similarity with us, with eukaryotes. Our membrane is bacteria type, but our DNA duplication system is archaea type. And we can make a long list of things that are a chimera all over the place. And in 1992, Carl Woese written, the archaea related to us, to the eukaryotes. They are descendants of the microorganisms that gave rise to the eukaryotic cell billion of years ago. In his idea, we had Luca, we had a division that brought the bacteria and the archaea, and then at the base of the archaea, things divided again, and you get the eukaryotes and the archaea. The tree of life, the three domain of life, bacteria, archaea, eukaryotes. This is what you study in school all the time, right? Well, we have new evidence that really changed it completely. We should rewrite textbook right now. Because a few people working in deep sea hydrothermal sediments, they found a bunch of microorganisms that when you do archaea, when you do their phylogeny, you look for protein that we call eukaryotic signature protein. If normally you have one or two of them in archaea, this guy, they're all over the place. Look at this. They're very similar to the eukaryotes for this. And they call this group of guys Loki archaeota. They're now within the Asgard archaeota division. Yeah, exactly. But the point is that if you do these phylogenies, and this is based on DNA extracted in the environment. We didn't have any organism with us. We're doing this as a reconstruction of DNA from the environment. This suggests the eukaryotes, the proto-eukaryotes, the original cell that then got the mitochondria and the chloroplast, is not from the base of the tree, like many people artificially believed earlier, but it's actually a branch inside the tree, which means we don't have a three domain of life. We have a two domain of life, bacteria and archaea, and then they merged to create the eukaryotes. Now, this is not just a big change in terms of where we come from, it's a huge change in the time we interpret everything else in the evolution of eukaryotes. And after 10 years of work, a Japanese group, Kentakai and Hiroichi Imachi, they were able actually to isolate one of these archaea from sediments, 10 years to grow it, when I told you that things grow slow, usually well beyond the lifetime of a PhD student. And they were able actually to isolate these very strange looking archaea that is in symbiosis with the methanogens, and they propose a pathway by which this guy with these strange appendages might have got interaction directly with a proto-mitochondria, an alpha protobacterium. And today, this guy, which is called Prometeoarchaeum, other reference to alien aficionados, I don't know how many are out there, but this might be the way eukaryotes started. So in a way, we are more microbial than we believed. The phantomatic proto-eukaryon out there might have been an archaea. This small snapshot of what is happening after Luca is just to show you how complex things are with things that we still have in our hands. We can sample bacteria, we can sample archaea, we can sample eukaryotes, and yet we have a huge debate happening on how things are related, not just where they come from. And there's a lot of work that needs to be done in this sense. Now, there's one thing you should consider all the time. What we know and what we lost. Microbes don't live, for the majority, take away stromatolites and little else, a few smudge of carbon here and there in some rocks. They don't leave fossils, right? So it's difficult to know what we lost over time. So what we do, the famous top-down approach, which is what I worked on, while Joseph worked on the bottom-up, and I'm sure he spoke with you about reverse TCA cycle, catalytic network, has been, okay, let's take genomes, and let's look into genomes, and let's try to understand when things started. So this is a work by a colleague called DuPont. They took all available genomes at the time, and they did phylogenies, and they tried to see where different binding sites for metal, which are a proxy for different metabolism, started. And well, as you look at this, everything looks like started very early in the evolution of life. So the suggestion here is that life started and very soon became able to do everything, occupied all the niche, was very fast in colonizing the planet and taking over everything. Now, this is a view because we don't know what we lost, right? So we tried a similar exercise. We said, well, we don't know microbes well, but we know isotopes and we know rocks. Let's go inside rocks and let's look for proxy that tell me which one of these metabolisms was present. And very quickly, all the metabolism appears to have appeared with uncertainty, which is this dashed line, very early in the evolution of the planet. And the limit here is not that we don't have evidence earlier, it's that we don't have rocks earlier. We don't have that many rocks that are older of 3.8 or 4 billion years. So we cannot keep further, but it seems like everything started very early. And if you actually do the top-down approach of doing phylogenies of all the organisms and try to see where things are, so you see this is a tree of life, bacteria and archaea, not eukaryotes, it will be complicated to place them, with the dating of the emergence of the major groups. And then you try to line up the isotopic evidence of when a metabolism started. And then you see that, for example, the yellow dot here, which is sulfur oxidation, is all over the place, but it doesn't match the evolution of any of the groups. In other words, we know that the metabolism started somewhere, but we don't see anybody emerging as a major group there. We do the opposite here. We know when land, soil started to be built, and at the same time we find fossil of plants, and we can relate the two. We know where things started to fly, and we find fossil that fly. And we do the same with photosynthesis. Well, with metabolism, we cannot really do it. It's like we lost the signal. Okay, these guys are doing all these things, but these things do not line up with the emergence of this guy. What this suggests, that either we don't know anything of what we're doing, or there were a lot of lineages that got lost. A lot of groups of bacteria or archaea or organism overall that were once doing things, probably they're the one that got the innovation first, that they got lost. So what we know and what we lost is perhaps the biggest question we have in biology right now. And the only exercise you can do is take today donor, electron donor and acceptor in the environment and look for the different metabolism, photosynthesis, methanogenesis, aerobic respiration, which is what we do here. And then you can try to place constraint on when electron donor and acceptor were available and when not. And then you can try to play back the tape and say, okay, this was 2.5 billion years ago. This was 3 billion years ago. This was 3.8, as old as we can go. So we think these were the possible metabolism at the time. But again, this is due to a very incomplete picture of things we have. And you can play this tape up and down and think about these things, but we still have a very incomplete picture. Now, to close off, and then we have time for questions, what's my take on origin of life? What do I think about we're going to be able to do with origin of life? Well, you know the Miller-Urey experiment, right? You study it everywhere, not just in biology or in chemistry. Everybody studies it. Why? Because it was the first solid evidence that you can make biotic material from abiotic material, right? You can make organic molecules that are building block of life starting from inorganic things. Look at this. This is the original journal. Chemists add missing piece to the theory of life origin. This is early on. They got the Nobel for this. Now, when they done this experiment, the scientific community, the journalists, the lay public, everybody was sure we had found the origin of life. This must be it. We can do amino acid. And then years goes by, and we figure out that we can do amino acid in many other ways. Mineral surfaces, hydrothermal condition, they're in comets, they're in the interstellar medium. Finding amino acid is exciting, but it's not a proof that life is there anymore. And so is not finding sugar, or nucleosides, or lipids. So my point is that we found one way to do amino acid. We believe that was the way of doing amino acid. And now we know there are multiple ways of doing amino acid. And we have no idea which one was the original one, or if all of them happened at the same time. And with the origin of life, I believe we're going to do the same. I am convinced in the future we're going to cross the boundary between abiotic to biotic. We're going to be able to start life. And the moment we do it, we're going to be convinced that that's the origin of life. But as we've done in the past, we're going to probably discover multiple ways to cross that boundary and control that transition. Assuming it's a hard transition and it's not a continuum. That's another discussion we can have. But I'm pretty sure we're going to find it, we're going to cross it, and then we're going to find in the next 100 or 200 years multiple ways of doing it, and we're never going to know, short of a time machine, how life started on this planet. So I'm sure we're going to solve the origins of life. I'm not sure personally we will ever solve the origin of life on this planet. And so that's that. Thank you very much.